The Global Food Regulatory Science Society. GFoRSS mission includes knowledge generation and dissemination, underpinning food regulatory decisions, in support of food control systems. Consistent reliance on food regulatory sciences enables robust food control programs.
Good morning, good evening, everyone. I am Dr. Ruba, a food safety expert at JFDA, the member of the Global Food Regulatory Society. It's a pleasure to be moderating today's event, which is the second event organized by JFDA.
Dr. Ruba, your sound, your voice is very low. Please, if you can adjust it, please. Thank you.
Okay. Now, is it okay?
It's a little bit better. If you can increase it, it will be okay. Thank you.
Okay. Good morning, good evening, everyone. I am Dr. Ruba, a food safety expert at Food and Drug Administration, and a member of the Global Food Regulatory Science Society. It's a great honor to be moderating today's event, organized by GFoRSS as part of its Food Regulatory Science Laboratory. Welcome to you all. It's a pleasure to have participants in the event. And again, welcome. And let's begin.
Dr. Ruba, we still have the technical problem of the voice. So if you can, please fix it. Thank you.
Hello, can you hear me clearly? Can you hear me?
Yes, thank you.
Okay. So can we start?
Yes, but if you can increase your voice, thank you.
Okay. Good morning, good evening, everyone. I am Dr. Ruba, a food safety expert at Jordan Food and Drug Administration, and a member of the Global Food Regulatory Science Society, GFoRSS. It's a great honor to be moderating today's event, which is the second webinar organized by GFoRSS as part of its Food Regulatory Science webinar series. Welcome to you all. It's a pleasure to have participants from all around the world. So welcome again, and let's review the topics of the upcoming webinars. So this webinar will discuss the titanium dioxide safety assessment as a food additive, review of the joint FAO-WHO expert committee report. Titanium dioxide, a topic of immense interest following recent assessment by both EFSA and JECFA. So we are fortunate to have Dr. Mark Feeley with us today. Dr. Feeley is the former Associate Director of the Bureau of Chemical Safety at Health Canada, and brings decades of experience in food safety, particularly in chemical assessments. He'll be guiding us through JECFA's latest risk assessment on titanium dioxide and sharing insights on its safety as a food additive. The global regulatory landscape is rapidly evolving on titanium dioxide, especially after the European Food Safety Authority, EFSA, raised concerns in 2021 and made the decision to delist titanium dioxide from approved additives. This session will shed light on JECFA's methodology, key findings, and challenges in assessing titanium dioxide. Dear participants, please note that Arabic interpretation is available for this webinar, and kindly submit your questions throughout the presentation in the chat box, and there will be a dedicated question and answer segment after Dr. Feeley's talk. Dr. Feeley, we are excited to hear your insights, and over to you, please.
Thank you very much, Ruba. I guess from the beginning, I'd like to thank the organizers for preparing this webinar, and once again, thank you to Samuel Godefroy for inviting me to talk about titanium dioxide. There's been a few recent developments regarding the the approval and use of titanium dioxide in a number of products, specifically food additives. And the purpose of the talk or this webinar will be to provide an overview of the EFSA position, which was back in 2022 or 2021, and compare that to the recent JECFA update, which was completed in late 2023. And both of these assessments are pivotal to the continued use or not use of titanium dioxide as a food additive. So if, in order to get started, if I could have the presentation queued up. So just as Ruba has indicated, the purpose is to look at titanium dioxide. So we can scroll through these. So it's fairly unusual, titanium, as one of these chemicals that sort of was by accident discovered to have very good refractive properties. And by refractive properties, we mean that it has very good light scattering ability. So the largest use of titanium dioxide is as a pigment. And it was actually, we go back over about 105 years ago, that it was actually discovered how to manufacture titanium dioxide for use as a pigment. And it's one of these things that you read about and figured that, well, there can't be a lot of uses for titanium dioxide. However, since its inception as being able to be manufactured from titanium ore, there's been over 200 million tons of titanium dioxide produced, with probably about 90% of that titanium dioxide being used as a pigment. So it's used as a pigment, that anything that requires a white color in a number of products. So these are paints and plastics, enamels, paper products. It also has very large uses in personal care products, such as toothpaste and cosmetics. It's also a primary ingredient in sunscreens because it has a unique ability to absorb and refract ultraviolet rays, which would be harmful to human skin. So, but since its use or identification as a very pure white color, it also has extensive use in foods. So it's approved for use in a very large number of food categories. And in fact, because the original assessments identified no toxicity associated with its use, the use levels are according GMP, which is good manufacturing practice. So theoretically, any quantity of titanium dioxide that is deemed to be necessary for its purpose of use, which would be to impact a white color to the food, is approved. So the whole point of titanium dioxide is to brighten food products. There's also some potential use as an anti-caking process based on its granular or crystalline structure. And if you look at the wavelength of visible light, which is from 400 to 700 nanometers, the optimum wavelength of light that titanium dioxide imparts is usually between 200 and 350 nanometers, so right in the visible light range. And this allows it to impart the strong white color, in this case, to foods. So something which is unique and very applicable to titanium dioxide assessment, especially for use as foods, titanium dioxide crystals or particles less than 100 nanometers are basically transparent. So therefore, they would serve absolutely no purpose for use as a food additive, because they would not impart the white color which titanium dioxide is known for. So we can have the next slide. So as I mentioned, there's probably earlier uses of titanium dioxide before the food regulatory agencies actually caught up to industry practices of applying chemicals that had not gone through a full regulatory assessment, because titanium dioxide first entered into production back in actually 1916. So one of the earlier food regulatory agencies that approved the use of titanium dioxide was the US FDA. And not too long after that, the joint FAO-WHO Expert Committee on Food Additives, JECFA, conducted their original assessment back in 1969. So earlier assessments of chemicals to be used as food additives were generally not as robust as they are today. So simply the limited number of data that was required for the safety assessment of titanium dioxide was mainly predicated on the fact that in some limited human absorption studies, basically no increase in titanium could be detected. And therefore, the idea or the premise came out that titanium dioxide, when added to food, was basically very poorly bioavailable. And even based on the limited toxicity studies at relatively high doses, no effects were seen. So at that point, the JECFA decision was to set an acceptable daily intake of not limited. So essentially what they meant was that there were no toxicological effects associated with its use, and therefore we had no concerns. And that type of decision allowed Codex to assign GMP to this food additives. So essentially that meant that if industry deemed it useful to use titanium dioxide in any food categories, depending if the food was what particular category, titanium dioxide could be used. So essentially, the issue arose that a few years ago, probably about 5 years ago, this is the issue where nanoparticles started to become known, primarily thanks to analytical chemists who were able to develop the methodology to start measuring particles reliably at less than 100 nanometers. So in the time of the earliest toxicological assessments, it was estimated that less than 5% of titanium dioxide being used in food was in the nanoscale. So this is very important because at the current time, there was a greater interest from a toxicological basis of nanoparticles. So the recent decision by EFSA was actually based on a number of studies that started to occur and were publicized back after 2016. And they're in EFSA's most recent assessment. They concluded that the initial estimate where less than 5% of titanium dioxide being used in food was composed of nanoparticles. Their new assessment had indicated that there was a significantly higher percentage of titanium nanoparticles that were likely present in food-grade titanium dioxide. So, and why JECFA had decided to update its previous opinion was that in 2021, there was a new safety assessment published by Titanium Dioxide. And this was an update from their previous 2016 position where they had indicated they had no toxicological concerns regarding the use of titanium dioxide in food. So in the 2021 EFSA position, they included or considered a number of studies that were performed with pure titanium dioxide nanoparticles. And based on the results of those studies, there was some indication that titanium dioxide was involved in genotoxicity. And there's a number of studies and endpoints that they looked at, but primarily it was dealing with DNA damage and chromosomal aberrations. So there were a number of other EFSA considerations that were published in 2021 besides genotoxicity. They indicated they also had concerns with potential immunotoxicity, and this is based on the availability of titanium dioxide to gut-associated lymph node tissue, which is very prevalent in humans in the small intestine. They also had concerns regarding the ability of titanium dioxide to induce inflammation, so inflammatory reactions in the GI tract. And also, and this is something that we'll look at in a little bit more detail, is that they also considered that titanium dioxide may be involved in development of something known as aberrant crypt foci. So these aberrant crypt foci are lesions that develop in the intestinal tract and are considered to be precursors for colon tumors. So there is quite a bit of concern regarding that. The initial concern about the ability of titanium dioxide to produce aberrant crypt foci was actually developed by the French Agency for Food, Environmental, and Occupational Health and Safety. And that was in 2019, and that was based on a study that had been conducted where animals were exposed to titanium dioxide both with and without a colon tumor initiating agent, and basically they reported positive results. So, and what you will also see is that there's a IARC classification, so International Agency for Cancer Risk Assessment, that has indicated that titanium dioxide is potentially a human carcinogen. And you'll see this listed in a lot of publications and online review articles. However, what they fail to mention is that classification is specific only to titanium dioxide when inhaled. So they considered that there was insufficient evidence for any other exposure route except inhalation. So, and you would consider that there are other crystalline particles, silicone specifically, where if inhaled, they are integrated into the respiratory tract and cause a number of adverse effects. So because EFSA concluded that they couldn't rule out any aspect of genotoxicity, that precluded them from developing an acceptable daily intake, and therefore the use of titanium dioxide as a food additive in the European Union was withdrawn. And this is, we'll speak about this a little bit more when we get into the EFSA opinion, but the European Commission was very quick at ruling that titanium dioxide was no longer approved for use in food sold in the European Union. So we can go on to the next slide. So this is simply something that I borrowed from the School of Chemistry at the University of Birmingham in the UK. This just gives you sort of an indication, picture-wise, of what size of materials we're actually dealing with. So the titanium dioxide nanoparticles, so something that's considered a nanoparticle, generally has a size category in one dimension of less than 100 nanometers. So generally, nanoparticles are considered to be anything between 1 and 100 nanometers. So you see that things such as the carbon nanotube and the gold nanoparticle, these are all considered nano-sized materials. And there have been studies and experiments conducted with pure titanium dioxide nanoparticles in the very small range. So they're down lower than 5 nanometers. So these are chemically synthesized as pure nanomaterials. So we can go on to the next slide. So in the EFSA evaluation, the initial, as I indicated in 2016, EFSA did not consider that titanium dioxide raised any toxicity concerns. And this was based, even at the time, they knew they were issues with nanoparticles. They considered that at the time, the food grade titanium dioxide in Europe, which goes by the European registration number of E171, was not considered to be a nanomaterial. So the preliminary reports that were coming out that titanium dioxide, specifically nanoparticles, were causing toxic effects were not taken into consideration in 2016. However, in 2019, EFSA came out with new guidance regarding what exactly should be regarded as a product of nanotechnology, and considered that titanium dioxide nanoparticles were implicated in any subsequent safety assessment of food grade titanium dioxide. And this is about the same time that EFSA realized that their earlier estimate of the percent contribution of nanoparticles in food grade titanium dioxide was underestimated. So there's a couple of different factors that led EFSA to revisit food grade titanium dioxide, or E171. But primarily, I think what motivated them to do this reevaluation was the fact that they now realized, with additional analytical methodology, that the extent of nanoparticle contribution to titanium dioxide, food grade titanium dioxide, was significantly greater than they initially estimated. So there were a large number of titanium dioxide studies that had been conducted. Some of them were conducted with the food grade titanium dioxide, but a large number were being conducted with titanium dioxide nanoparticles. So 100% of the test material would be nanoparticles, and some of these were with nanoparticles less than 30 nanometers. So very small and pure nanoparticles. And what that showed was that these titanium dioxide nanoparticles could produce DNA damage, and more specifically, chromosomal damage. So there's an issue specifically with titanium dioxide nanoparticles and mutagenicity testing. So in a typical evaluation of a food additive, you have both mutagenic properties and genotoxic properties. And genotoxic properties usually refer to the ability of a chemical to physically damage DNA or cause breaks or deletions in physical chromosomal structures. For mutagenicity, these are stable, inheritable changes in genes. So one of the issues with titanium dioxide being able to produce mutations is that one of the main modes used to determine the mode of action is with bacterial cells. So there are standardized bacterial assays for detection of mutagenic chemicals. But what was noted very early on is that bacteria do not have the process to internalize titanium dioxide, even nanoparticles. So therefore, there was no ability for titanium dioxide in, in prokaryotes or bacteria to interact with DNA and cause mutagenic changes. But regarding this, the EFSA panel considered that the evidence for DNA strand breaks and chromosomal damage was significant enough that they could not rule out that nanoparticles present in food grade titanium dioxide was not a genotoxic risk, and therefore it could no longer be considered safe when used as a food additive. And it's sort of which is a prominent feature of the European Commission. They're very quick to pick up on EFSA opinions. So by 2022, the European Commission removed all authorizations for titanium dioxide in foods. So the coming into force date they gave, which is approximately 6 months after the European Commission regulation was initially published. However, this does not include the use of titanium dioxide in medicinal products. So anything that might be considered to be associated with pharmaceutical use of titanium dioxide has not been included in this ban. And EFSA is looking at revisiting this opinion regarding the approved use of titanium dioxide in medicinal products in 2025. So, and obviously the removal of approvals for a food additive that's used worldwide has caused some concern among other food regulatory agencies. And the majority of these who have conducted their own internal assessments subsequent to EFSA indicating they could no longer approve the safe use of titanium dioxide. And in particular, Health Canada, the Food and Drug Administration in the US, FSANZ in New Zealand and Australia, UKFSA, and more recently, JECFA, have not agreed with the EFSA opinion. And there's an asterisk by FDA because they are currently in the process of responding to a petition to ban the use of titanium dioxide, even though they have come out in support of the JECFA position. So we can go to the next slide. So as I mentioned, part of the large number of studies that EFSA revisited when they updated their safety assessment of titanium dioxide in 2021 was to look at titanium dioxide nanoparticles. So these are pure particles between 5 and 50 nanometers in size, and looked at their ability to cause DNA damage. So the overall position was that there were enough positive results that they concluded that these titanium dioxide particles, specifically nanoparticles, could cause DNA strand breaks and chromosomal damage. And there's a number of assays that look at that, both in vivo and in vitro. But one of the issues that has still been undecided, I think, is the mode of action of how titanium dioxide is able to actually damage DNA. And there are some studies that have shown that these DNA strand breaks or chromosomal damage that titanium dioxide nanoparticles are able to induce is associated with oxidative stress. So this is oxygen radicals that are produced through an inflammatory process. And the thinking is that that is at least partially responsible for the observed chromosomal damage that's being induced by titanium dioxide nanoparticles. Now, it's usually a fairly important issue, because when you start to induce DNA damage through a non-direct interaction process, such as inflammation or oxidative stress, these are considered to be events that are threshold. So theoretically, you should be able to observe doses where this does not occur. Anyways, the other issue that EFSA concluded was that in the experiments available, they could actually not identify a size category of titanium dioxide nanoparticles that was not associated with genotoxicity. And also, they could not conclude that the DNA damage being caused by titanium dioxide was involved in sort of only a threshold mechanism. So a mechanism where you could potentially identify exposure which did not cause DNA damage. So I think we can go to the next slide. So as I mentioned, in the original 1969 assessment that was conducted by JECFA, they primarily relied on the fact that there were both studies on a number of species, including some human volunteers, that not analyzing for titanium dioxide, but analyzing for titanium, there was no indication of absorption from the GI tract. And generally, when you see a food additive that is not bioavailable, unless it's producing immediate site-specific effects in the GI tract, it's generally considered to be non-toxic. And if a food additive is considered to be non-toxic, JECFA usually does not assign an acceptable daily intake. They consider it to be unnecessary. And that's essentially what they indicated, was that there was no issue with having to establish an acceptable daily intake. And this is back in 1969. And because there was no issue regarding toxicity, there was no specific dietary exposure conducted at the time. And the advice to the Codex Alimentarius Commission was that titanium dioxide could be approved for use according to good manufacturing practices. So the the update to that was that the Codex Committee on Food Additives indicated in 2021 that JECFA should reevaluate titanium dioxide. And this is primarily based on the EFSA conclusion that they could no longer consider the safe use of titanium dioxide as a food additive. So from a time standpoint, JECFA reevaluated titanium dioxide at its 2023 meeting. This included a large number of toxicity studies. And during this evaluation, JECFA also considered engineered nanoparticles. So these would be specifically pure test substances produced primarily 100% of titanium nanoparticles. So all sizes of titanium dioxide less than 100 nanometers. And these were included in the evaluation that JECFA conducted. They also considered that there were sufficient studies conducted with E171, and In the codex system, the food additive numbering scheme is INS, so International Numbering System, and that's also 171. So if you see any publication that says E171 or INS 171, these all deal with food grade titanium dioxide. So one of the main issues that JECFA considered in looking at some of the properties of titanium dioxide is that when you have pure engineered titanium dioxide nanoparticles, so these have a very narrow size distribution range, and they, based on the size distribution range, they have different physical chemical properties, and they are not considered to be representative of food grade titanium dioxide or INS 171. So I think this is an important distinction, that JECFA considered that there was sufficient studies that had been conducted with E171 or INS 171 involving dietary exposure for them to conduct their assessment. But more importantly, they considered studies that had been conducted with only pure titanium dioxide nanoparticles not to be representative of what was being used in food. So we can go to the next slide. So this is just some of the INS 171 or food grade titanium dioxide particle size distribution as determined by transmission electron microscopy. So there's relatively detailed analytical methodologies that is required to get a size distribution of particles generally considered to be representative of food grade titanium dioxide. So you see that particle size, and this is based on food grade titanium dioxide that has been supplied by the Titanium Dioxide Manufacturing Association. Also, there is analysis of food grade titanium dioxide that has been deposited with FDA in terms of use as food additives, which is part of their food additive approval process. Manufacturers have to provide the material that they intend to add for food, and FDA does their own analysis. But so essentially what you see is that the black line represents sort of the mean of the particle size distribution, even though there is a fair amount of range. And if you look at the 91st percentile range distribution, you get to the average particle size in food grade titanium dioxide as being slightly less than 100 nanometers and going up to above 250 nanometers. And this is something that would be expected considering titanium dioxide particles less than 100 nanometers serve no purpose in terms of light scattering and producing a white color associated with its use as a food additive. So one of the things that the clever chemists were able to show is that the inter-lab variability is quite great. So if you compare labs, you see that's responsible for the gray area. Those are inter-laboratory comparisons, whereas the between sample variability conducted in a lab is relatively consistent. It's only when you compare lab to lab that you see some of this variability start to come out. But the other main thing that has been identified by all food regulatory agencies is that there currently is no harmonized method available for extraction and characterization of titanium dioxide particles after it's been added to food. So you see that, and once again, this is food grade titanium dioxide. The median particle size, so at the D50, which is diameter 50, is ranges from about 104 up to about 160 nanometers, which is what you'd expect if you want to produce a white color. And at the lower end of the range, there's only about 11% of particles by mass that would be considered to be nanoparticles, so less than 100 nanometers. So we can go to the next slide. So as part of the JECFA assessments, they looked at available studies, once again, both in experimental animals and humans, to see how much titanium dioxide is bioavailable. And in a well-designed and conducted animal experiment, they considered that titanium dioxide from dietary exposure, they confirmed that the absorption generally is very low. So, and this very small number is based on the analytical limit of detection. Now, if you look at titanium dioxide nanoparticles, the potential bioavailability is higher. It's usually approximately about 0.02%, and in certain cases can range up to about 0.1%. But once again, JECFA's position is that studies conducted with pure titanium dioxide nanoparticles were not totally representative of food-grade titanium dioxide, or INS 171. And I think in the variety of human studies, which look for bioavailability of titanium dioxide, JECFA came to a very important conclusion, considering this food additive has been used for decades, that if it was bioavailable, you might expect to see higher concentrations of titanium when you assessed a large age category of humans. And this they did not see. So there are human studies looking at titanium levels in blood and tissues, which spanned age ranges from late 30s, early 40s, up to late 70s, early 80s. And basically, there was no consistent increase in titanium seen across these age categories. So this gave JECFA confidence that the potential bioavailability of titanium dioxide in humans was very, very low. So one of the earlier studies that had been conducted and which JECFA reevaluated was a National Cancer Institute study specifically looking at titanium dioxide. The material used was a pigment known as unitane 0-220. And what was conducted by FDA, and the results were available to JECFA, was it was considered that this unitane 0-220 compound was essentially equivalent to INS 171. So that meant that this study that had been conducted by NCI, where this unitane 0-220 was added to the diet and fed to experimental animals throughout their lifespan, was applicable to the safety assessment of INS 171. And essentially what we see at very high doses, so these doses are 2.5 grams per kilogram body weight for rats, 7.5 grams per kilogram body weight for mice, fed over a lifetime, produce no consistent toxicological effects. So JECFA also looked at the large number of mutagenicity and gene tox studies that were available specifically for INS 171, or food-grade titanium dioxide, but indicated they could provide no convincing evidence for the possible genotoxicity, specifically of food-grade INS 171, or titanium dioxide. But one of the issues that has been raised is that the current methodologies for assessing both genotoxicity and mutagenicity aren't specifically adapted for nanomaterials. So there's a current large studies going on to try to define the specific test studies that could be applicable to the assessment of nanomaterials. So I think, and we'll discuss this study even a bit more, but the one study that EFSA actually commissioned was an EOGRT study, which just stands for Extended One Generation Reproductive Toxicology Study. And EFSA had initially said earlier that this study was not available for them to review. So this study was commissioned, and the results were available both to EFSA and JECFA. And basically what it concluded that in looking at extended exposure to titanium dioxide resulted in no developmental or reproductive effects in these animals. And these are at relatively high doses over the course of the study. So up to, a gram per kilogram body weight per day over the lifespan. And what was deemed acceptable was that there were no effects on any generation for reproductive toxicity. So we can go on to the next slide. So, as I mentioned previously, the French Food Safety Authority had evaluated a study which indicated that food-grade titanium dioxide seemed to be able to promote precursors of colon tumors, and these are aberrant crypt foci. So these occur in the colon, and in certain cases, in humans, they have been shown to progress to colon tumors. So in this particular study conducted by Bettini et al., when food-grade titanium dioxide, in this case E171, was administered to the animals in drinking water, they demonstrated an increase in these aberrant crypt foci. So the guide of the studies was that they both showed an increase in aberrant crypt foci, both with and without the treatment of a colon tumor initiating agent. So the particular study, usually what you do is you expose an animal to a colon tumor initiating agent, and subsequent to that, you give a chemical that you think might promote the formation of colon tumors. In this particular study by Bettini et al., what they reported was that these aberrant crypt foci developed both with and without the colon tumor initiating agent. So subsequent to that, studies have been conducted where E171, or food-grade titanium dioxide, was administered in the diet at higher doses. The dose where ultra-crypt foci were shown to be forming in the Bettini et al. Were only 10 milligram per kilogram body weight per day. In the subsequent studies where titanium dioxide was administered in the diet at much higher doses, no increase in aberrant crypt foci were observed. So what this likely represents is an issue related to how the animals have been exposed to titanium dioxide. In particular, following the report of increase in aberrant crypt foci, additional studies have been conducted with E171, and the majority of these studies involve either exposure via drinking water or by gavage, which prior to dosing, the solution is sonicated in order to break up any aggregates or agglomerates that might be developing. So when you add a pure titanium dioxide powder to a solution, what you generally observe is agglomeration followed by aggregation and sedimentation. So the agglomeration tends to be loose areas where particles have formed larger particles. And generally what happens is that these large particles significantly increase the size and diameter of the titanium dioxide. But by sonicating those aggregates or agglomerates, this breaks them up to their constituent nanoparticles. So what was discussed at the JECFA meeting was that even though there were additional studies that report GI tract effects, so these are all changes in the colon which could potentially explain why these aberrant crypt foci developed. These were primarily done with studies where there was some sort of physical disruption of the titanium dioxide prior to dosing the animals. So JECFA considered, recognized, even though these results did indicate a possible mode of action, they were of questionable relevance to the safety assessment of titanium dioxide in food, which I think there was a report put out by Health Canada where they did a state of the science assessment of titanium dioxide. And they essentially came to the same conclusion because they queried industry about how titanium dioxide was added to food. And industry reported that there was no physical disruption processes being applied, which is consistent with the use of titanium dioxide particles less than 100 nanometers have basically no coloring effect. So industry reported that when we add titanium dioxide to food, we want to impart a white color, and that white color only occurs if you have particles of significantly larger sizes than 100 nanometers. So these large number of studies that are also available, JECFA indicated that because titanium dioxide nanoparticles have size distribution, and with that size distribution, they have physical chemical properties that are inconsistent with INS 171, they indicated these studies were of somewhat limited relevance to the safety assessment of food-grade titanium dioxide. And we can go to the next slide. So as I indicated, when you have food-grade titanium dioxide and it has a relatively size distribution where the particles are greater than 100 nanometers, this would have different physical chemical properties than a dosing which was made with pure titanium dioxide nanoparticles. So, and more importantly, what JECFA realized is that the methods to produce these titanium dioxide nanoparticles were inconsistent with the same manufacturing methods used to produce food-grade titanium dioxide, or INS or E171. So the other important point that JECFA raised was that any study conducted with food-grade titanium dioxide, either E171 or INF 171, which are essentially the same thing, should be able to adequately assess the potential toxicity of the titanium dioxide nanoparticle fraction. So based on this, and the fact that they confirm that titanium dioxide does have a very low bioavailability, they reaffirmed their 1969 ADI of not specified. So essentially that means that the food industry can continue to use titanium dioxide at GMP. And in fact, I think the Codex General Standard for Food Additives, the GSFA, which is a numerical list of all the uses and levels that food additives have been approved for by Codex, lists over 60 categories of food that titanium dioxide continues to be approved for use in. And the other important detail is that JECFA also updated its food additive specifications. So basically, JECFA meetings deal with both the toxicological aspects, including the exposure aspects of a food additive, but they also deal with the manufacturing and chemical aspects of a food additive. And essentially, in order to be approved for use, a food additive has to follow JECFA specifications for manufacturing and purity. So essentially what JECFA did was to describe a manufacturing process which, if followed, produces titanium dioxide, which has generally a particle range between 200 and 300 nanometers. Now, in reality, the particle size distribution in food grade titanium dioxide allows for smaller particles, but once you get into issues involving agglomeration and aggregation, these particle sizes get larger. So even though the particle sizes in the pristine food additive are likely smaller and outside the 200 to 300 nanometer range, by the time they're used in food, they increase in size. So I think this next slide, if I'm not mistaken, provides an example of that. So what we see here, this is a pure engineered sample of 6 nanometer titanium dioxide nanoparticles. So as I mentioned, once you put these in any sort of solution, what you immediately see is agglomeration, which is sort of a loosely defined collection of particles forming, and then subsequent aggregation, which involves a stronger and a denser assembly of these particles. Plus, you generally tend to see some sedimentation throughout the solution. So these titanium dioxide nanoparticles are in straight phosphate buffer. So they go in as a pristine sample of crystalline titanium dioxide nanoparticles of 6 nanometers in size. And what happens is that based on aggregation and agglomeration, you see the particle size that ultimately forms when you look at the right hand, where the median diameter of the particles changes from 6 nanometers up to over 200 nanometers. So this is what's likely forming in foods when you add food grade titanium dioxide. You get these agglomerates or aggregates forming that have the pure white refractive index that you want as a food additive, but also increase the size beyond the nanoparticle range. So we can go to the next slide. So these are just some brief points that I've included for information. And these detail some of the main studies that have been conducted with INS 171 or E171, which essentially is the same thing, or a test material considered to be equivalent to food additive titanium dioxide. And in that case, as I mentioned, JECFA considered that this pigment that was used by the National Cancer Institute in the US involving unitane 0 to 220 was representative of titanium dioxide. So the overall conclusion was that high dose exposure of this chemical in diet provided no evidence of carcinogenic effects. And these are relatively high doses. Specifically, they were the high doses tested. EFSA also considered that in terms of providing evidence for a carcinogenic effect, the results from the study were negative, but it also indicated that the study was not appropriate for specifically assessing the potential for titanium dioxide nanoparticles to produce a carcinogenic effect. So there's the distinction is that EFSA considered all of the titanium dioxide test results with nanoparticles, which they had indicated was producing genotoxic results. And the study with this unitane compound did not provide them any convincing evidence that a similar study conducted only with titanium dioxide nanoparticles would have produced a positive result. So the next major study, which has been conducted with, in this case, E171 or INS 171, is the study that I mentioned with Bettini et al. And this was initially reviewed by the French Food Agency. And what happened is that both treatment with and without a colon tumor initiating agent, this is the DMH, the dimethylhydrazine, which is a well-known colon tumor initiating agent in rodents. Treatment of the E71 produced these aberrant crypt foci, and that led to a large number of studies, not only trying to understand how, but also to indicate was there something specific to the dosing. So there is a specific protocol called the nanogenotox protocol, where in order to fully assess the potential for nanoparticles and to get around this agglomeration and aggregation aspect that happens with pure ultrafine nanoparticles, the dosing is basically put in a dispersal agent, in this case 0.05% bovine serum albumin, but also ultrasonicated prior to dosing. So that allows the nanoparticles to maintain their size. So this is an important aspect, I think, when it comes to assessing the potential toxicity of food-grade titanium dioxide. Even though it does contain a fraction of nanoparticles, through these processes of agglomeration and aggregation, these nanoparticles as a percent contribution to food-grade titanium dioxide are likely significantly reduced. So JECFA considered this Bettini et al. Study, but they also looked at other studies where either the titanium dioxide had not been ultrasonicated and put in a stable solution, such as BSA, prior to dosing, or that the titanium dioxide had been added to the diet. And these studies show no effect on the ability to induce altered crypt foci. So I think this is a relatively important point that does point towards an issue how the dosing mechanism can produce an artifact of a toxicological study. So, I mean, EFSA did recognize that the aberrant crypt foci were likely being produced by E171 because the test substance had been pre-dispersed and stabilized in a media. So they also indicated that it's unlikely that if you had a pure solution of titanium dioxide nanoparticles, that it would provide negative results for this ultra-crypt foci type of assay. So we can go on to the next slide. So this is the this was the study that was commissioned, and it was identified by EFSA that there was no definitive reproductive toxicity study that had been conducted. So, and this has just recently been published by Battersby et al. In 2024. So the food titanium dioxide was administered in feed. So once again, diet. And this is over a number of generations. So it generally starts with the animals being exposed through sufficient reproductive cycles, and it goes into breeding to produce an F1 generation. The F1 generation continues on exposure to the test substance. They are also then bred to produce an F2 generation. And through this study, they can also look at a variety of other endpoints, such as immunotoxicity, neural behavior, but also in this particular study, they also looked at dietary exposure to titanium dioxide and its ability through transgenerational exposure to induce these aberrant crypt foci. So, as I indicated, there was exposure for 10 weeks prior to mating, continued exposure all the way up to generation of F2. And what it showed, what there was no toxicity in the animals, no developmental, and no reproductive effects. So the only thing that JECFA noted was that there was a minor issue with some developmental immunotoxicity assay. However, they did not consider this to be significant enough not to assign the no observed adverse effect level at the highest dose tested. And this developmental immunotoxicity study results primarily was related to the pure response or poor response based on the positive control. So, and this is expected to be repeated. However, EFSA, in looking at the same study, indicated that while there appeared to be no effects on reproductive toxicity, they were not convinced that this study is reflective of the particle size distribution of titanium dioxide when it's added to foods. So, and this is interesting because it's also been recognized that there is no validated method to extract titanium dioxide particles after it's been added to food and do an adequate size distribution. So this is an issue that the analytical chemists are currently dealing with, because generally to extract titanium dioxide from food, there's an acid treatment and physical disruption required, which would break apart these agglomerates or aggregates that could be added. So EFSA does still have some concern that the test substance, known as E171 or food grade titanium dioxide, any experimental results with that could be representative when it's added to food, because there's been no size distribution of the E171 after its addition to food, which really is an analytical method that has to be resolved. However, the overall outcome of this, including the results from these specific studies, is that JECFA considered that there were studies that had been conducted with E171 or INS 171, where it's primarily being added to a food matrix, which is its main use as a food additive, and considered that these were sufficient to assess any particular toxicity from titanium dioxide nanoparticles, specifically the size distribution in food-grade titanium dioxide. So essentially that led JECFA to concur with their previous position that an ADI was not required for titanium dioxide. And subsequently, JECFA reported back to Codex that there was no requirement to change its current food additive approvals. And these are all listed in the Codex General Standard for Food Additives. And we can go to the last slide. Oh, I thought we had nanoparticles there. Anyways, that's it. Thank you very much for your attention. And I guess we can look at any questions that might have come up.
Thank you. Thank you very much
Ruba?
for this detailed presentation. And I would like to remind everyone that the presentation would be available on GFoRSS website. And also, again, they can use the chat box or they can raise their hands and ask their question. So I would like to start with my question first, if possible, and then we will go to the raised hands. Given the difference between EFSA and JECFA's conclusions, how should regulators approach titanium dioxide in the national food safety standard? You know, as you know, part of us is regulators here who are attending the webinar.
Well, I think that's a good question, Ruba. I would, for food regulatory agencies, I think it really comes down to, do they have a specific guidance regarding the assessment for potential toxicity of nanoparticles in general? So this is what precipitated the EFSA reevaluation, because they updated their policy regarding nanoparticles. Originally, they had identified that if a test substance was being manufactured and it had less than 50% of nanoparticles, it didn't fall into their nanoparticle regulatory environment. So if your food safety regulatory agency has not developed a specific policy for nanoparticles, I would say there's nothing to preclude the use of the JECFA position, which is specific and in accordance with a large number of other food regulatory agencies have looked at this data. Essentially, specifically when you're looking at food grade titanium dioxide, manufactured according to the updated JECFA chemical specifications, I would concur that there would be no issue with the use of titanium dioxide as foods. So a country that is looking at its general internal risk assessment and regulations, I think you would concur with JECFA. However, a food manufacturer who wants to export their product to the EU, unfortunately would have to follow the European Commission, which basically has banned the use of titanium dioxide. So you would have to either look for an alternative or change your specification for your food product and remove titanium dioxide.
Thank you very much, Dr. Feeley. And I would like to start with Yasmin Ahmad. I hope she's still with us. Yasmin Ahmad. We can't hear you, Yasmin. Yasmin, can you switch on your mic? I think we can move to Sayyid Muhammad al-Matari, maybe, until Yasmin is on with us. Mr. Muhammad al-Matari. Okay. Then we will go to Redha Krishna. Hello. Your mic is off. We have a lot of raised hands, but I think they are not responding. Farah Naji.
Hi, everyone. Can you hear me?
Yes, we can.
Okay, so thank you very much, Dr. Feeley, for your great presentation. It is a very good learning. I might have a question when it comes to an alternative or replacement to titanium dioxide in our foods. I understood from your presentation it can be either used as a pigment, a colorant, or even as an anti-caking agent. So, for example, if I'm putting it intentionally as a coloring agent, is there a specific alternative or a replacement that we can actually refer to that will give me the same result as the concerned ingredient? Thank you very much.
I mean, actually, that is a very good question because, I mean, there's all sorts of other anti-caking agents that don't require a crystalline product such as titanium dioxide. So there should be, if you're using it strictly as an anti-caking agent, which I think would be minimal, because the majority of use of titanium dioxide is as a pigment. Now, unfortunately, I'm not overly familiar with what alternatives industry is practically looking at. I've heard there's an extract from whole rice kernels. So you've after the rice has been milled and you have white rice, there's an attempt to extract the colorant responsible for that white rice pigment and use that as a pigment in place of food. However, just based on the type and the light refractive, it's not going to be an exact match because it's one of those things where titanium dioxide has the entire market for white colors in foods. I mean, and that's why you have a separate titanium dioxide manufacturing association who are very interested in its use. So, unfortunately, I'm not overly familiar, but I know, if you're specifically looking at generation of a white color in foods, and this I would think you would only need to do if you were exporting a food to the EU, because they no longer allow for food uses. I think it's likely that the Titanium Dioxide Manufacturing Association might be coming up with alternatives, even though they will continue to indicate that the use of titanium dioxide in foods is safe. So I guess a long answer to your question is that outside of potential constituents of hulled rice to impart a white color to food, I'm not overly familiar with other possible alternatives, because titanium dioxide is still, even though it's still being used worldwide with the exception of the EU as a food coloring agent, it is still being used food-wide as a coloring agent in all sorts of pharmaceutical products. And once again, it has a very important application for use as a sunscreen, because there's really at least limited alternatives to something it's in its ability to refract or deflect UV rays, which would be the primary purpose of a sunscreen.
And I would like to invite, I don't know, Astro Nadiai, so excuse my pronunciation. Yes. You have to put your mic on. Hello?
Hello.
Hello. Can you hear me?
Can I speak in French? You can.
I will translate. You can speak French. You can
You
speak
can
French. Yes.
translate? Huh?
Yes.
It's not
We will
possible.
translate. Go ahead.
Yes.
No, we will translate. Go ahead.
Thank you. Bon, moi, c'est Astou. Je suis dans un laboratoire au niveau du Sénégal et je crois que ces additifs sont très utilisés dans nos aliments et les produits cosmétiques. On aimerait quand même que le JECFA nous dise la limite acceptable pour l'utilisation, que nos pays, ils ont une réglementation un peu faible, donc il faut qu'on se base sur des études scientifiques comme le JECFA pour pouvoir limiter, les industriels puissent limiter l'utilisation de ces genres d'additifs dans les aliments, parce que nos enfants en consomment beaucoup au niveau scolaire. Et les études ont montré que c'est cancérigène. Donc, vraiment, par rapport au One Health, il faut que ça soit vraiment international, que des limites de ces genres d'additifs puissent être fixées par les scientifiques. Voilà. Donc, voilà, moi, je travaille sur la sécurité sanitaire des aliments depuis plusieurs années. Mais en tout cas, les additifs, c'est l'oxyde de titane et le glutamate monosodique, des gens comme ça. Il faut vraiment qu'on nous dise la limite acceptable par rapport aux études menées et qui montrent que c'est cancérigène. Il faut qu'on soit limité. Les industriels puissent nous faire des produits alimentaires aptes à la consommation, qui ne menacent pas
Merci,
notre vie.
Astou, merci. Je vais traduire, s'il vous plaît. So I'm going to attempt to quickly summarize the question posed by our colleague from Senegal. So indicating that these additives have a wider use right now in a number of applications, including in food, and basically asking what is the conclusion after all? What is there an acceptable limit set by JECFA? Are these or is this additive really cancer causing? Simply to be able to reflect that in the regulatory system of developing countries, which tend to follow the JECFA assessment and Codex assessments.
Thank you for the question, and thank you for the translation, Samuel. I thought I was back in a bureau director meeting or something. It's a good question. I mean, certainly, it's one of those issues of a food additive that has very wide uses, and this is what you might expect when Codex indicates there's no toxicology concern with a food additive and it can be used at GMP. And I think when I looked previously, their titanium dioxide could be used in up to 60 different food categories at various concentrations. And these concentrations are set by industry, presumably to achieve the required technological effect, which would be to impart a white color. So, and as you mentioned, they're in pharmaceuticals, they're in personal care products, they're in toothpaste, and more importantly, they're in pharmaceuticals such as pills. So a lot of pills, they impart a white color based on the use of titanium dioxide. Even EFSA, I think, in recognizing that coloring agents aren't really considered to be essential from a food additive perspective, did not ban them, at least currently, in any pharmaceutical products. So from that aspect, I would consider, especially based on the fact that the pharmaceuticals need that white color, that there is basically limited to no risk from exposure to titanium dioxide in pharmaceuticals. And I think the study, the best studies to date, especially for a chemical that's representative or a test substance that's representative of food additive titanium dioxide, even at very high doses, there is no indication that titanium dioxide represents a carcinogenic risk. So in general, I would say that even though there is exposure from a large number of products, not just food, but from other products, especially a lot of toothpaste use titanium dioxide. I would consider that based on these very low levels of exposure, that there is limited concern when it comes to them representing any sort of carcinogenic risk.
Thank you very much, Dr. Feeley. There is a question in the chat box from one of our GFoRSS. What is the best method for measuring titanium dioxide? Is that the ICP-MS or is it the ICP volumes?
So what
is the best method we can measure the titanium dioxide in food?
Well, yeah, that's thanks to the chemists. I mean, if you want the best method, it's probably transmission electron microscopy, which isn't exactly a well-known method or applicable method that a lot of analytical labs have available to them. Now, this is specifically if you want to measure particle size distribution. If you want to measure titanium dioxide in foods, my suggestion is that currently, if you're not worried about particle size distribution, so you're not looking at nanomaterials, that you can just use something like some mass spectroscopy. So, even something like a standard metal screen like ICP-MS, which will measure titanium, because the majority of titanium you get in food is already in the oxide form. So if you just wanted to see, and usually there's a very good correlation between food indicating it has titanium dioxide as a food additive and the measurement of titanium. So any specific method to do metal measurements would be sufficient to pick up titanium. If you're specifically interested in measuring titanium dioxide and nanoparticle distribution, then you're going to need some additional special mechanisms associated with your mass spectroscopy. If you don't have the availability, which a lot of analytical labs don't, for doing transmissible electron microscopy or even scanning electron microscopy. So a lot of the particle size distribution can also be picked up by mass spectroscopy, but that requires things like single particle inductively coupled plasma tandem mass spectroscopy. So something relatively more complicated than standard ICP-MS. So it really all depends. If you want to verify that a food is not using titanium dioxide as a food additive, then I would say just do a titanium measurement, and ICP-MS should be sufficient.
Thank you. And there's another question from a GFoRSS member. If EFSA is making regular safety evaluations on additives, and when you can find evaluation results on additives you find
Can you repeat that, Ruba? I didn't, your audio was breaking up.
Is EFSA making regular safety evaluation on additives? And when we can find evaluations results on additives used combined together?
Okay. Well, the prior to the formation of EFSA, which I think is a very good organization because EFSA actually has to respond to a large number of food regulatory agencies in the European Union. So any country in the European Union can request that EFSA produce an updated opinion for a food additive. In this particular case, there was a large number of food additives that were, let's say, grandfathered in prior to the formation of EFSA. So EFSA was looking at them on a priority basis to do reevaluations. And they're very good at publicizing when they're revisiting food additives and publishing the outcome of those updated food additive positions. So they did that in 2016, then they did it again in 2018, and then finally in 2021, their most recent opinion was formed regarding the potential safety of titanium dioxide and indicating their recommendation is that it no longer be used as a food additive. Now, the question about countries that might do assessments on multiple food additives at the same time is also a good question, because whenever regulatory agencies look at food additives, it's always in isolation. Unless you have a specific concern that a food additive might be associated with a particular medical condition in humans, you wouldn't necessarily include exposure from other food additives. So that's something that, you know, in some cases, regulatory agencies actually look at that, similar to the issue with, let's say, artificial sweeteners that contain aspartame, and which is produced from phenylalanine. There's an inborn genetic mechanism where some humans cannot process phenylalanine. So any food additive containing a phenylalanine moiety is considered in the safety assessment of a particular food additive. And that's sort of an example that's few and far between, but the majority of food additive assessments tend to be done in isolation and not considering combined exposures.
Thank you very much, Dr. Feeley. And to wrap up today's discussion, we highlighted the complexity of assessing titanium dioxide as a food additive and JECFA's reaffirmation of its ADR status, contrasted with EFSA's machinery stance underscores the need for more comprehensive data, particularly in human epidemiological studies. Thank you once again, Dr. Feeley, for your experts' insights, and to everyone for your participation. We hope this discussion helps regulators and food safety experts navigate the evolving science and entrepreneurial next. We hope to see you all at our next webinar on the 15th of this month, where the subject will be developing the Thank you. Thank you very much, everyone. Thank you. Pleasure.
All right. Well, thank you very much. And once again, many thanks for the Global Food Regulatory Science Society for organizing this. And special thanks to Samuel Godefroy for inviting me to participate.
Thank you very much.
All right.
Thank you.
Bye, everybody.
Take care.
Bye.
The Global Food Regulatory Science Society. JFoRSS mission includes knowledge generation and dissemination, underpinning food regulatory decisions, in support of food control systems. Consistent reliance on food regulatory sciences enables robust food control programs.